Good to be with you again this morning. Thank you for the opportunity to speak. And it's been a special joy to get to know Jerome and his wife, and what a marvelous job he's doing on internal evidence. I know it's been a thrill to all of you to participate in that. So we make a good team. We're looking internally and now at this hour externally to why we should believe in the great God we serve. I mentioned yesterday at the beginning that my purpose in this series is to help all of us have a deeper appreciation for the magnificence of the God who made us. And if I can accomplish just a little bit of that, if you're a believer, that you leave a stronger believer. And if you're not a believer, that you're going to at least examine this more deeply, because there's much to be learned from God's book of the natural world. So thank you for being here. Much more could be said, but we need to get at it. I forgot to get my clicker here ready, so let's get that out. I bring my own clicker, class, because I've run into situations where clickers don't work, and that only ruins my whole lesson. So I bring my own clicker. Our topic for this hour is the great designer, evidence from the chemistry of living things. You live, ladies and gentlemen, in a time where we've learned more about how living things function than ever in the history of mankind. In fact, I'll make it stronger than that. Since 1950s, some of you are alive in 1950s? Yeah, thank you. It did exist. Yeah. Since the 1950s, we have learned more about how life functions than in all of the history of mankind prior to that. I want to give you a couple of illustrations. In 1953, Francis Crick and James Watson discovered the structure of DNA, for which they received the Nobel Prize in 1962. Complete change in what we understood. About that same time, Frederick Sanger sequenced the first protein. That protein was insulin. And Frederick Sanger sequenced that protein. Nobody had ever done that before. That was 1952. He received the Nobel Prize for that. And interestingly enough, Frederick Sanger went on to sequence the first DNA also in the 1960s. And he received another Nobel Prize in chemistry. Nobody gets 2 Nobel Prizes in chemistry, but Frederick Sanger did. That all started in the 50s, class, in the early 60s. So there are a host of other things taking place as these things unfold before us, and man is just becoming overwhelmed with what we're learning about how life functions at the biochemical level. And man was also becoming haughty as a result. A lot of things were coming about in those days that people were saying, the more we learn, the less we need God. Because we're learning where they don't need God. And of course the accusation is you just stick God in where you don't understand. It's the God of the gaps. And I'm here to tell you, class, and I hope this is very clear by the time we finish, that what we're arguing in this series of lessons is not God based on the gaps or God based on our ignorance. It's based on the more we learn, the more powerful is the evidence that there's a God. Not based on gaps, but based on knowledge. One other thing I wanted to mention about that same time, there was an object put up in space in 1957. In fact, I have the date here. I better get my glasses out because I can't read it. July the 16th through the 24th. Well, no, this is October the 4th, 1957. October the 4th. Anybody remember? Sputnik went up. You children know what Sputnik is? It was the first object put up in space by what nation, everybody? The Russians. By the way, if you weren't here last night, I told the audience that the elders approved my teaching this like a class, which means you can answer my questions. Sputnik scared us all to death because we knew the Russians were going to take over space and rule the world. And one of the things that happened in the 1960s was that the government passed a whole bunch of laws about increasing financing for science education. And I'll have to tell you, I was one of the recipients of that largesse, because they gave Harvard University 50 scholarships one year, which the government financed. And they said, you get science teachers and you train them to be better science teachers. Harvard chose to pick one student from each state in the union to come to Harvard that year to get a master's degree at their expense. I got the one for the state of Florida, folks, and got to go to Harvard in 1967, 68, get a master's degree at their expense. I've told you already, I'm only telling you this to say I have lived through this I'm talking to you about. It's personal. So I was involved in the Chem Study Curriculum Project. How many ever heard of Chem Study? Nobody. Thank you very much. It only reorganized the entire chemical curriculum, that's all. College and high school teachers got together and rewrote the curriculum. They did the same in biology with BSCS biology. They did the same in math. You remember the new math, parents? Our parents hated it because they didn't understand it either. But it was a method of trying to get people to understand math, not just learn how to do it. I'm not saying it was completely successful. But those were days of change. It was a remarkable time, folks. You have lived through a remarkable century in terms of what we've done with the sciences. And Sputnik motivated a lot of that. And of course, we caught up with them, didn't we? And the date I mentioned before, July the 16th through the 24th, 1969. What happened, class? That's right, thank you. The first man to land on the moon, Neil Armstrong, Buzz Aldrin, and Michael Collins, Apollo 11. And they landed on the moon prior to the turn of the century, turn of the decade, as President Kennedy had predicted. And it was a great triumph for the United States. A little sidelight there just for your information. Neil Armstrong and Michael Collins both became stronger believers having been there. Buzz Aldrin, on the other hand, became a stronger atheist. Same evidence, different results. So don't think that because we're going to pile on the evidence, everybody's just going to change their minds. People see them in different ways. But I'm telling you, the God of heaven has so opened up the floodgates of information to mankind, it is difficult for me to conceive how anyone can dismiss this and not see there had to be a designer. But about that time, Time magazine came out. This is August the 8th, 1966. Is God dead? Look, this all fits together. The evidence from science was multiplying. People think we're just learning so much, it's going to be just a matter of time where we know not only how life works, but how to make life. And it was during that same time that the song you just sang was written by A.W. Dicus, 1966. I will tell you, it was a direct response to this movement. And I loved it that you sang with great vigor the chorus. One time, I wanted you to sing it one time, and you did it powerfully. And what did you say? Our God, He is alive. In Him we live and we survive. I also asked him to sing verse 3, which says, secure is life from mortal mind. God holds the germ within His hand. Though men may search, they cannot find. For God alone does understand. I had him lead that because in the 1960s, we had a professor at Florida College who refused to sing that verse. He was our biology teacher. I'm pausing. Why would he refuse? Because our professor in biology at our college, and we are a Christian college, believed that mankind soon would learn how life works and we could make life ourselves. So he did not believe what verse 3 says. So he refused to sing it. And what I'd like to tell you is this. I wish that man were still alive. Because in the last 50 years, folks, we've discovered so much more about life at the biochemical level, and we are more lost than we've ever been as to how in the world life ever came about without a designer. There is no way life came from non-life by itself. That view is absolutely destroyed by the evidence we've produced in the last 50 years. Of course, people are still trying to figure that out and think they can do it. Such faith I've not seen in all of Israel. But I'm saying to you, there were times when even our brethren who were informed scientists thought we may not need to have God to explain life. Sorry about that. But I'm telling you, for that same professor and whoever else is in his court today to say that same thing, well-informed, would have a great more difficulty defending his view. Here are some of the things that were said in that magazine. J.J. Altizer and William Hamilton in their book, Radical Theology and the Death of God. Folks, these are Methodist preachers who were quoted. We must realize the death of God is an historical event, that God has died in our cosmos, in our history. And the dead God is the God of the historic Christian church and beyond the church of Christendom at large. So we've watched in our generation, they said, that God died. Now, how in the world is that possible? That's possible if you made him up in the first place. So these folks came to believe that man made up God, and of course that's what a lot of folks think. Nature's all there is. And nature produced us, and we, out of our intelligence, decided there was some kind of God. So that was being proposed rather strongly in those days, and I will tell you as a college student at Florida State and Harvard University, and later the University of South Florida, I had professors in the sciences who would clearly say to us, you folks who believe in God in this class better get over it, because it's going to be but short order that the need for God in any dimension is going to be overruled by the sciences. So, I like this book better. And I have it with me. Is Atheism Dead by Eric Metaxas. I highly recommend it to you. I mentioned one yesterday, God's Crime Scene. You remember this one? I highly recommend that. This is another good one. Eric Metaxas is a good writer. He's not a science guy, but he's a good historian and he's a good writer. And so you need to read this. Because in this book, copyright 2021, he makes this statement. Since the Time article in 1966, because this is a takeoff on that, roughly 5 things have arisen to challenge, and I will argue, overturn the secular consensus that formed in the wake of that article. I like the way he says that, because that's what happened, class. There was a secular consensus that kind of took over after that. Because God's dead, so we're going to look to the sciences as our answer. He says there have been 5 things happen since then that overturn that thing, he says. And I would agree. And here are his 5 things. He said, science has now taught us there's a beginning. Did I say that last night? Now, we didn't talk about it very much. But there's a lengthy description of that in here and also in Meyer's books that I mentioned to you. And by the way, I'll mention that again while we're talking. I forgot to mention some books last night, but this is his most famous book, Signature in the Cell. You need to read this if you're going to dig deeply. He's excellent. And I want to kind of comment on what Jerome said a while ago about recommending authors. I too will make an excuse for myself. I'm not recommending everything people say. You get that, I hope. But there's some things they say that are powerful toward the evidence for the existence of God. That's what I'm after. And here's the other one I forgot to mention last night. It's a new read for me, The Fortunate Universe. Fortunate. Luke Barnes, the second author on here, is the student of Geraint Lewis, who's the teacher. They're both Australians. And this book is a discussion between those two men back and forth. Luke, a believer, Lewis, not. And it's about fine-tuning. And it's a little heavy, but it's a wonderful read about why the evidence for fine-tuning is powerful. Because frankly, Luke Barnes knocks down all the arguments against it. I recommend that to you. All right, and then he says, fine-tuning, which we did a treatment of last night. And I went home last night and prayed God's forgiveness for the weakness of my presentation. Because we didn't touch the hem of the garment, folks. Did you hear me? We didn't touch the hem of the garment. And then, stupefying complexity. I told you in the first lecture, I want you leaving here stupefied. And I mean it. What we've learned is stupefying complexity. He's absolutely right about that. This morning and tonight, that's going to be our topic. And then he said archaeological discoveries. Maybe you didn't know this, but there have been a lot of archaeological discoveries that support the text of the Bible. You may be dealing with that later, I'm not sure. But even recently, things that support the historicity of Scripture. That's not our topic for mine. And then what atheism is and produces. We've learned what atheism does. Folks, the 20th century was the century of atheistic governments. The Nazis and the communists, both of which are founded on godlessness and man being the answer. And they have destroyed us. I think I have a reference here I want to read you. Let me see if I can find that. It's out of Metaxas' book, and these chapters are worth reading if you don't read anything else. Because he's talking about the pure evil of atheist regimes. The blood of those murdered in the name of atheistic totalitarianism cries out for justice. And if we cannot at least acknowledge these unavoidable facts, what does that say about us? It is ironic and nightmarish fact that in a very short time during which atheism was adopted by entire nations, it stands alone in history as guilty of the very things that the modern atheists accuse us of. You know what like the new atheists like to do is talk about how many people, how many horrible things the church did in the name of Jesus. And folks, it is the case that about everything you can imagine has been done in the name of Jesus, including wars, torturing people to death, the Inquisitions, lots of horrible things. But Metaxas argues in this text, and I'll argue it too, anything that was done in the name of Christ doesn't touch top side or bottom of what atheists have done. And they simply ignore that, the new atheists. In the year 1937 alone, dedicatedly atheistic Soviets murdered 106,300 clergymen. The Soviets, in the course of their adoption of what they called scientific atheism, murdered between 12 and 20 million human beings, most of them their own people. Let us say it again. Nowhere, even among the most egregious examples of religious malfeasance in history, do we begin to approach the scale of such atheistic atrocities. It is atheism alone, and especially scientific atheism, to use the cheerful Soviet parlance, that sought to murder in mass those they could not tolerate and with whom they did not wish to coexist. And why not? If you believe the atheistic theory of how we got here, there is zero reason for you not to kill off anybody you don't want around. And that's what they did. The last thing we need is an atheistic government. And that's been demonstrated in the last century. So, class, are you ready for chemistry? All right. I'm going to start with Romans 1 again because that's our theme passage from Scripture. His invisible attributes are understood by the things that are made, Romans 1 says. What can you understand? His eternal power and his divine nature. He's powerful, and he's not part of this world. Something outside. And that's the argument from design, which is what argument we're using. We are looking at things that are made. I'm looking at God's other book, class. So welcome to Biochemistry 101. Thank you for joining my class today. It's our first day of class, and I'm welcoming you. And I want to lay a few ground rules. This class is very important for your future. If you're going to be in the science area at all, you must grasp all of it. All of it won't be on my tests, but a lot of the important things will. So I expect your rapt attention. I do not allow sleeping. We'll do something about it if you start sleeping. I know you've just been through another lecture, and you had a night's sleep, I hope, or maybe you didn't. And I meant to mention a while ago, you got to bring your sweetheart with you. So glad you did. She's gorgeous and makes your compliment. I couldn't bring my Marilyn this time. She just wasn't up to it this time. Better for her to stay home. But I'm bringing greetings from her. I was on the phone with her for an hour yesterday, telling her all about you. And she sends her love and greetings. And she's the love of my life. So pardon me for that little side trip. And that had nothing to do with biochemistry. Well, it did, too. It really did. We have 4 kids and 16 grandchildren. And she is the master mama and grandmama. In this class, ladies and gentlemen, we will learn about the biochemical basis for living things, which to me is the most passionate and important thing we'll learn. Much of what was learned has been learned in the last century. So we begin. This is our topic. Let's talk about how life is structured. So we're going to use Francis Crick in his little book, Life Itself, Its Origin and Nature. If you were listening a while ago, class, Francis Crick's a famous scientist. He is the co-discoverer of the structure of DNA, 1953. If anybody knows about the biochemical basis for life, it's Francis Crick. And so he's going to tell us about its nature, what is life like, and secondly, its origin. How did it get here? That's a pretty hefty thing for such a small book, don't you think? So I'm turning to page 72 as our beginning, and as we do, we'll look at our screen up here that kind of summarizes how every living thing functions. Life as we know it on Earth appears as a synthesis of 2 macromolecular systems. And I just lost half of you. Every living thing, class, has 2 systems of giant molecules. Every living system. You with me so far? The proteins, because of their versatility and chemical reactivity, do all the work, but are unable to reproduce themselves in any simple way. So, Here are some proteins, class, in living things. There are the structural materials made up of proteins that make up the construction, the shape, and mobility of living things. The things that involve shape and mobility are generally proteins. And proteins, class, are assembled from amino acids, which we'll talk about more in just a moment. But they're proteins. That's one great class. There are also tools and machinery in every living thing. We usually call them enzymes. They're just special forms of proteins. So both of these are proteins, class. Say it. Good. I'm asking you to say that because that's the answer to most of my questions over the next few minutes. And I expect you to answer me when I ask you a question. I'm telling you the answer. Now, it's not all the answers, but it's most of them. So we have these two sets of materials in every living thing, and both of these are proteins. But there's another class. The nucleic acids seem rather tailor-made for reproduction, but can achieve rather little else compared with the more elaborate and better equipped proteins. So there's another class of molecules, which you call the blueprints. That's the nucleic acids. We call them DNA and RNA, and they are good for reproduction. They are for the information and directive function of living things. And I'm going to say something that Dr. Crick says, so don't blame it on me. I'm reading from the book. He says the RNA and the DNA are the dumb blondes of the biomolecular world. Sorry, blondes. I didn't say that. Fit mainly for reproduction with a little help from the proteins, but of little use for much really demanding work. Not very nice. So we'll forgive Dr. Crick. But the fact is, these, without these, don't do much. And these proteins, without the nucleic acids, can't reproduce. And if you're a living thing, class, you not only have to function, you have to be able to reproduce yourself. Every living thing. So, the problem, he admits on page 72, of the origin of life would be a great deal easier to approach if there were only one family of macromolecules able to do both jobs. But life as we know it employs two, always. Did you get that? That's the first question on the quiz tonight when you get back here. I'm kidding. But if you were in my class, tomorrow you'd have a quiz. This is important stuff. Living things have to have both. That makes describing the origin more difficult. So now I'm going to jump ahead here because I'm afraid I'll forget it later. I'm getting old. On page 88 of this same book, he says, an honest man armed with all the knowledge available to us now, and by the way, this was published in the 1980s. We've learned a whole lot since the 1980s. But class, we very well knew most of what I'm going to tell you this morning by the 1980s. So it's old information in that sense. But we knew nothing about any of it before the 1950s. An honest man armed with all the knowledge available to us now could only state that in some sense the origin of life appears at the moment to be almost a miracle. So many are the conditions which would have had to have been satisfied to get it going. And I say, amen, except for the almost. The plain fact is, he goes on to say, the time available was too long, the many microenvironments on Earth's surface too diverse, the various chemical possibilities too numerous, and our knowledge and imagination too feeble to allow us to be able to unravel exactly how it might or might not have happened. In other words, we don't have a clue. But there's possibilities. And then I want you to listen to this last paragraph of chapter 6. If it was highly likely for life to have evolved from non-living things. That's what he's talking about. Highly likely? Are you kidding me? Then, of course, there's no problem. But if it turns out it was rather unlikely, then we are compelled to consider whether it might have arisen in other places in the universe where possibly for one reason or another conditions were more favorable. Did you catch that? By this time, we're already saying the earth is 4.5 billion years old. He's saying to us maybe 4.5 billion years wasn't enough time to get all this accomplished here. So what do we have to do? Look somewhere else in the universe. And folks, I'm not kidding you. The rest of this book is about directed panspermia. And you don't know what that is. Panspermia is the idea that somebody from outer space seeded the earth with the first life forms. Either naturally or some civilization sent it here. He has a whole chapter on what the spaceship might have been like that sent the first life forms here. Class, do we know one thing about panspermia for which we have solid scientific evidence? Zero! But this Nobel Prize winner chemist would rather have that than God, for which there is zero evidence, but we'll take that over God. So that's what he does for the rest of this book. He talks about directed panspermia. Absolutely blows my mind. And he ought to know better. So here we are, class. We're back to our study. I told you that proteins are made up of amino acids. So we begin our biology class, our biochemistry class, with the 20 common amino acids. I say that because these are common to all living things. If you take a protein out of one of your cells and a protein out of a single-celled E. coli, it has the same amino acids in it. Did y'all know that? All living things are made up in their proteins of these 20. Now, I've been told by medical students, I don't know if my dentist back here would agree, there are a few exceptions to that, aren't there? That's what I've been told. But, folks, it's good enough for government work. I mean, this is 99.99% of all the proteins in all living things everywhere on earth. They're made up of the same stuff. Now, I want to interject here. How do you explain that? That's one explanation. But there's another one. The evolutionist says the reason we have a common chemistry in all living things is because we are all from the same tree of life. We evolved from the simplest forms into all of us. And of course, out of that comes all the common chemistry. That is not a stupid answer, class. That is a possible answer to this fact. I will ask you one thing. Don't argue with facts. You're going to lose every time. It is a fact that the chemistry of life is unified. All proteins in all living things are made of these 20. So how do you explain it? Well, as a believer in God, I think I would say this. It's perfectly reasonable that a designer would make a system of living things that have to interdepend on one another out of a common chemistry. That is perfectly reasonable, and you don't have to start over every time you make a new living thing. You could use a common framework. It's a very reasonable explanation. If you want to know which of those is the best, you go digging deeper. But both of them are reasonable explanations from a scientific standpoint, in my view, though most scientists would reject the God hypothesis because they think it steps outside of science. May I take another little side trip here, class? You do have to step outside of science to propose God, because you can't investigate God with science, because he's not part of the natural world. And science deals only with the natural world. Get that? So let's just admit, science can never lead us directly to God, because you can't use science to investigate a non-natural being. But can you use science, class, to point to all kinds of evidence in the natural world that somebody did this or something did this? Yes, you can. And so you can conclude from that there must have been a grand designer. which is the way we go about it. All right, back to the amino acids. This is question number 2 tomorrow. I want you to be able to give me the names of the 20 amino acids: glycine, alanine, valine, leucine, isoleucine, proline, serine, threonine, aspartic acid, asparagine, so on down to histidine. That's not hard, 20. You can figure it out. It would be nice if you'd also memorize the 3-letter abbreviations. So glycine is GLY. That's sensible, isn't it? Most of them are very sensible. Alanine is ALA. Valine is the first 3. Isn't it the first 3 letters almost all the way? I'm missing it. Where am I missing it? Right here? Isoleucine, yeah. This is ILU. That's ILU. Anyway, ideally, you'd know the 20 common amino acids and their abbreviations by tomorrow morning, question 2 on your quiz. Why is that important? Because these make up all the proteins. That is only the most important substance in your body in many respects because it does almost all the work. Every protein is a combination of these 20. Well, I told you. A few exceptions, but mainly. It's very much like the 26 letters of the alphabet, which make up our words in English. Every word in English is some combination of those 26 letters. That makes it easy, doesn't it, to understand? So you got a string of amino acids, and a string of amino acids forms a class. All right, I told you the answer. A string of amino acids makes a protein. Exactly right. And these are the ones that make up the string. Okay, that's why their abbreviations are important. So, we'll come back to proteins in a minute, but I must talk about the other great class of molecules. That's the DNA and the RNA. Very brief. Again, I've already apologized to God for the brevity of this discussion. We should have spent a whole lot more time on proteins, and certainly a whole lot more time on this. But DNA, the structure for which Crick and Watson are famous, is a helix, a double helix with crossbars. You see this strand right here? And there's another strand wrapping around it. Can you see that? And then there are cross pieces. In this picture, they color them. Can you see that red always joins up with yellow, and green always joins up with blue? See that? That's because there are 4 bases along that strand of DNA. And I think I have the names here. Let's see. This provides it a capacity to reproduce. It's adenine, thymine, guanine, and cytosine. Those are the 4. And they go along this strand here, and they connect up. And it's adenine joins up with thymine, and guanine joins up with cytosine. A with T and G with C. Like love and marriage and a horse and carriage. That's question 3, class. A joins with T, G joins with C, and that's the way they match up. So, up here, the blue always joins up with the green, the red always joins up with the yellow. If the red happens to be on this side, then there's a yellow on that side. See? Class, do you have any idea why it's always that combination? It's because of the chemistry of the amino acids. And specifically, in this case, the chemistry of those bases, because they allow them to combine molecularly and make bonds. All right. Now, how does it allow us to make copies, reproduce? Well, because there's a language in there. This always blows my mind. When I was in school and they were teaching me about this, people were teaching this as if, well, isn't this interesting what we have discovered? And the question you ought to be having is, how is it that inside your cells in the DNA there's a language? Is there any evidence, class, of a language arising without intelligent source? Not that I know of. Language comes from an intelligent source. How did we get language in every cell in our body? We have it. It's called the genetic code. It's a language with 4 letters. The 4 I've just told you. thymine, cytosine, adenine, and guanine. And combinations of those 4 letters are formed into 3-letter words in this language. Every word has 3 letters. So you see before you on the screen, ladies and gentlemen, all the words you can make with 4 letters taken 3 at a time with repeats. I'm letting that sink in. If you were really in my class, I'd make you do the math. How do you know how many letters or words you can get taking 4 things 3 at a time with repeats? That's a math problem. Here's how you do it. How many can go in the first position of those 3 letters? 4, all 4 of them. How many can go in the second position? All 4. How many can go in the third position? Any of the 4, right? So it's 4 times 4 times 4. So we want to do the arithmetic? No fair using a calculator? 4 times 4 is what, class? And 4 times 16 is 64, and there are exactly 64 letters up there. Words. So now I'm going to teach you how to read. So here we go. Everybody's eyes up front. What if you had, let's go back. What if you had T as your first letter, T as your second letter, and T as your third letter? That's the word T-T-T. It's pronounced t-t-t. This is an unspoken language, ladies and gentlemen. But if you did pronounce it, it would be t-t-t. And right next to that on this chart, you see the letters P-H-E. Do you recognize that? If you don't, we'll go on. Let's try another one. How about A-T-T? At-t. That's illy. I-L-E. Does that help you? Not really. Let's see. How about C-A-T? Where is that? Right here. We can pronounce that one. That's cat. But it's really hiss. Has anybody gotten this yet? Those 3-letter in the parentheses are the abbreviations for the 20 amino acids that you just memorized, which we're going to have on the quiz tomorrow. So what you have here, class, in this language is a code for the 20 amino acids. But there's 64 words and only 20 amino acids. So that means you have some duplicates. Do you see repeats in there anywhere? Let me go back here. Yeah, leucine has 1, 2, 3, 4, 5, 6 different codes. Why does leucine have 6 different codes? Don't ask me. I don't know. But it looks to me like a backup system. You have more than one code in the amino acid. So several of them had more than one. Like here's threonine, 1, 2, 3, 4 threonines, and alanine, so on. So, but all 20 amino acids are represented here. In addition to that, if you look up here, you see there's 3 that say term, T-E-R-M. That is not one of the amino acids. What do you think term means, anybody? The end. It stands for terminator. It means stop. So there's a code in this language that tells you to stop. Now, it's not up here, but I can tell you also that the code A-T-G, this one right here, which means methionine, but it also means start. Everybody's eyes on me. What you have in this code, class, is a code for every one of the amino acids, and a code to start, and a code to stop. So you see that DNA strand? You can unzip it. It exposes 2 sides. When you read down this side, you're reading a bunch of 3-letter words that are made up of A, T, G, and C. And when you read down that line, you're going to find that the 3-letter words you read are these coded words. So if you come along and you come to the signaled ATG, it says, start reading. And so you start reading. And you read 3-letter words. Suppose the next one is TTT. That's phenylalanine, class. That means your first amino acid in that protein is phenylalanine. So you add it on. Then it goes to read the next three-letter word. Let's say it's ATC, that's threonine, or ACT. So threonine is added to phenylalanine, and you now have two amino acids in your protein. Are you with me, class? You look like you're glazed over. You're making a string of amino acids. Okay, so you keep adding one at a time, amino acids, and once after you get finished, you'll have a whole string of them, and then somewhere you're going to come along the code that says, stop! So you started reading, you read a bunch of three-letter words, and then you stopped reading. Those three-letter words stand for amino acids, so you've just built a three-letter word string, or a string of amino acids, which is class A. Whew, you're really weak. Between the start and the stop, you have a string of amino acids. That makes a protein. Exactly. Class, if you were a jury in a court of law and I was trying to convince you somebody designed this, I would say case closed. You have in every cell in your body a system that codes for building every protein in your body, of which there are thousands. And your body just builds them using that language. That is what we started discovering in 1953. Now, we didn't uncover all that I've just told you until about the 70s. But over that time, we were learning more and more about how things work. But that's nothing. Because all we've done there is just talk about how you get the code off of the DNA and make a strand of amino acids. That copy you made up here is a strand of amino acids coded, which is eventually going to be used down here to build a protein. But there's a whole lot more to this story. It's not just that you have a language up here in the DNA that you've learned how to use and read. We now know what it's doing in a large measure. There's still stuff we're learning, folks. Don't believe me when I say we know everything. We don't know the hem of the garment. But we know a lot. So my next job here is to go through with you just a little with how you build proteins. And I'm telling you, you first have to transcribe it. Go back. Well, let's go on to this one. Let's blow it up a little bit. This is the nucleus. Here's the DNA strand, and see here it's unzipped. And you're making a copy of it, and that copy is called messenger RNA. There's also over here a place where you're copying and making ribosomal RNA, and over here you're making transfer RNA. We don't even have time to talk about those. That's another 6 lectures. We'll wait. We're just going to talk about messenger RNA over here that is copied. And then you get this messenger RNA, and then notice it has to be matured. You see what's happened here? These 3 white areas have been snipped out and reorganized, so you now have a mature messenger RNA. We didn't know anything about that till the 70s. But that's what happened. And I'm coming back to that in a minute. And then the messenger RNA that's mature starts migrating. That's the next thing that has to happen. It has to migrate from here, copying it out. It has to go outside the nucleus to the factory. You have to move it. Let's go to the next slide. So here it is coming out of that hole right there, and it goes down here and is picked up by 2 ducks hugging each other. Doesn't it look like 2 ducks? It doesn't look like that, class. This is a picture. It is a ribosome that they made look like 2 ducks. But they're picking up this messenger RNA and reading the code. You see these 3-letter words here? They're reading the code, and with that code, they're producing a protein, a string of amino acids, which is a protein. Right. Class, does that look like a plan to you? Like somebody designed it? It sure does to me. I want to back up here. And if you don't get anything else today, you get this. Crick said you have to have the nucleic acids and the proteins or life doesn't work. One of the reasons that's the case is because you can't even get proteins without having both. You can't get nucleic acids without having both either. We're talking right now only about proteins. So I want you to get this, class. The code for the proteins comes from the DNA. The code is tied up in here. You have to unzip it to be able to copy the code. Guess what it takes, class, to unzip DNA? Thank you, that was very weak. I told you the answers. What do you think it takes to unzip DNA? Protein. Exactly right. It takes several proteins just to get it unzipped. Then you can make the copy, which is the messenger RNA. I'm going to show you a video of that in just a minute. But once that copy is made, here it is. You have to mature it. That means you got to snip out these. Those are called introns. Those little white areas, they're called introns. They have to be snipped out and reorganized to make them mature. Guess what it takes, class, to edit that RNA? Protein. Several proteins. There are really complex proteins called editosomes that help edit. And there are spliceosomes that help put it back together. Those are both proteins. If you stick in my class through 6 weeks, we're going to thoroughly study spliceosomes and editosomes. They're some of the most complex proteins you'll ever see. Made up of thousands of parts just to snip off that and edit it. And may I take a little side trip here, class? When we first started discovering this, one of the first reactions of evolutionists was, well, look at that. Here you've made a copy and you got to snip it out and edit it before you can even use it. No God would do that. Why don't you just make something that'll just be ready to use when you make it? Why make it so you got to snip things? That's stupid. I can read you from chemists that say those very words. And there was a whole movement from the 1980s all the way up into the 2000s where they called that junk DNA. Because they thought these introns that are snipped out are just useless. They're not used for anything. They're just snipped out and thrown away. It's garbage. God doesn't do that kind of stuff, they would argue. So there's no God. DNA doesn't support God. You see what I'm saying? When that first started coming out as a student of this material, I didn't know how to answer that. Because I don't believe God makes junk, do you? Or stuff that you just throw away. But I would say to people, and I did once to a professor who challenged me on this, and I didn't know anything I was talking about, but I said, sir, you might be a little more humble and say, we just don't know yet what's going on. And may I say to you, class, getting ahead a little bit here, there's now a book called The Myth of Junk DNA, because we're learning thousands of things that supposed junk is doing that we knew nothing about before. So be careful what you call junk. The fact is, those little guys that are snipped out are doing all kinds of fascinating things in your body, including making little RNAs that do all kinds of stuff that help you survive. In addition to that, class, please get this. If you have to snip out and reorganize, doesn't that give you more possibilities from that same strand? Did you know there's some living things where the same strand of DNA right there can make up to 1,000 different proteins by reshuffling and organizing it around? With those editosomes and spliceosomes, from the same set of information, you can make up to 1,000 different proteins. That means, class, there is more information stuffed in DNA than we ever dreamed, even 10 years ago. But back to the original. You've got the edited RNA. I told you it takes proteins to make this happen. It took proteins to make this happen, right? Not only did you have to have the DNA, you had to have a bunch of proteins. Okay, so now you're migrating and you're coming out here to that hole right there. When you get to that hole, do you think there's just any respectable hole in a nuclear membrane is going to let anything go through there? The answer is no. So what do you think guards the gate? Exactly. When that messenger RNA migrates up to that hole, it says, here I am, and the protein says, well, okay, welcome in. And he opens up like this, and the messenger RNA goes through, and he closes back up, and you have a protected nucleus. But that required on both sides of that hole. All right? I should have made you keep track for me in math. Are you good in math? We're too late anyway. But we've already added up a bunch of proteins, haven't we? Needed for the process. Okay, go. Next slide. Here it is migrating. Now it comes up here to the ribosome, and it's picked up and read like a tape through a tape player, and it reads these three-letter words. Then, class, it calls for the appropriate amino acid. GUC is one of the amino acids, or GUG. So go get me isoleucine. I don't think that's right, but go get me one. Well, you have to have a transfer RNA that brings that specific amino acid up here to be used to make these proteins, because you can't make them without the material. So you better have amino acids floating around in your cytoplasm. You listening to me? You got to eat the proper food. So you get a transfer RNA bringing up that specific amino acid. There are 20 of them. Class, there are exactly 20 specific transfer RNAs for those 20 proteins, which you also have to have. In addition to that, class, there are specific proteins associated with each one of those pairs that are also required. That's 20 more proteins. You got that? You're adding up, right? And so now you're getting down inside here, and you're bringing these in, and you're adding up one at a time. They connect up by particular molecules, valine and so forth, connecting up with each other. And when you get the final one, how do you know when to stop? Because there's a code for that says stop. And then it releases the protein. And you've just built a protein. Class, to make one protein by this method requires at least 50 distinct, separate proteins. So let me make it clearer. I'm going to go back up here. One of the original proteins up here that causes it to unzip and other things is ribonuclease. That's an enzyme. That's a protein. Ribonuclease, class, has to be built by this same process. You listening? So how did the first ribonuclease ever get built in a cell? Because it has to be in the very first step to build any protein. The same is true for all of the other of the 50 or so proteins needed in every step of the way. Where do you get them? By the same process. But each of them had to be built first, right? But how did you build them? Why do you think Crick would say, so many are the conditions that would have had to be satisfied, that it's almost a miracle? Folks, what does it take? A 2x4? So now I want to show you a little video clip. Because one of the problems with any of these presentations is you don't get a realized look. And this video does a good job. So there's a guy named Drew Berry, who's a biomedical animator. He works at the Walter and Eliza Hall Institute of Medical Research in Melbourne, Australia. He's amazing. If you want to have fun, go to his website and just look at several of his videos. They're incredible. So the one I've chosen to show you today is the one on protein building. And I have my master IT guy in the back who's going to control this whole thing for me and make no mistakes. All of you need to please thank John for me, because he's been a tremendous help already. So John, I'm ready to start our video. Stop right there. See there? Is he perfect or what? I command and he does it. I don't want to do that from up here. So this is the start of a gene for a bone strength protein. You're about to watch how a bone strength protein is built. So here is the strand of DNA right here. These things that are hooked to it are all different colors. They are not different colors, class. The animator colors them to make them look different, so you'll know they're different molecules. So let's count them. That's a different one, that's a different one, that's a different one, that's a different one. There's one, in fact, that's another one there. So 1, 2, 3, 4, 5. There's 5 different proteins already riding along the DNA strand. All necessary. Okay, let's proceed, John. As you watch now, there's going to be another little glob of protein join them. Whoops, stop right there. Thank you, John. You were ahead of me on that one. See this little guy up here? Go a little farther. Just a little, right there. That's vitamin D, class. Floating around in your nuclear cytoplasm. It's very important because that little guy is going to activate this protein right here. And when it does, the whole process begins. So get out in the sun and get your vitamin D, class. You want to build lots of good proteins. All right, let's go. Here comes the vitamin D, and you'll notice miraculously, stop, it changes that protein from red to green. It does not do that, but it does activate it, and green means go, doesn't it? So that's why it does that. Okay, so you've just activated this glob of proteins, of which this is the key, by the vitamin D, and now we proceed. Gene activation by vitamin D. And then we are joined by another glob of, what do you think, class? Yes, stop right there. There's another glob of proteins that have just joined, and you see the dark blue one inside here? That's the RNA polymerase I mentioned to you a while ago. So watch now, as the gene transcription begins, you got to now copy the information off the DNA. So here we go. You're going to see the RNA polymerase, stop right there, separates from this other glob. This is excused, this is still here, but folks, all of those were important to get this process started. And then the RNA polymerase guides it from there on. So it's going along here, go ahead. And we're going to wait just a little bit here. Right there, stop. The DNA is this strand. This is the RNA, that's the copy that's being made. You notice how it's coming from down inside here? This is the protein glob that is receiving all the stuff and making it into a messenger RNA, which is a copy of the information on the DNA. You're all with me, right? Thank you. All right, go ahead. Now we're going to go down and close in just a little bit, making it bigger in just a second. Transcription. Right, stop. Yeah. See all this stuff on the outside? This is all floating around in your cytoplasm. Very important because these are the pieces that go to make up the RNA. You got to have them. So they all migrate inside here and they make that thing start. So here we go. Let's now open it up and let's watch what's happening. All right, down inside there, can you see how it's unzipped? Stop right there just a second. You already did. Stop. Okay. All right, let's go a little bit further. There you go, right there. Do you see the code here along the DNA? GGT, AAG, and so on? Well, there's a G right there. When you make the RNA, the G always joins up with a C. So there'll be a C over here to join up with that G. A T is over here, so you get an A over here. An A is over here, so you get a T over here. No, you get a U, but don't ask me about that. T is replaced by uracil in the messenger RNA, and that's a complex subject. But the fact is, it's a mirror image copy. All right, so can you see what we're doing? We're making a copy of one letter at a time. Boom, 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 boom. And as we do so, we're making a mirror image copy of the information here on the DNA. That's what's happening. Now, what has been a problem in the past is these videos do not show you how rapidly this really takes place. So Drew Barry's goal in his video was to show you reality. So I'm seeing some of people's eyes are doing this, and I can't have it. I'm almost finished, but I don't want you to miss the rest of this, so you got to stand up for just a minute. I mean it. Stand up and shake yourself. I'm not having any sleepers. I know it's been a long morning. Too bad. All right, thank you. Nobody can leave. Have a seat. I want you alert for this. Eyes on the screen. What you're going to see now by Drew Barry is how fast this really happens. So go. Too slow. Too slow. Move it. Now that's more like it. You see that? Stop right there. Class. You can build a protein with 10,000 amino acids in microseconds. There is no human factory on the face of this earth that can touch top side or bottom of what's happening in your cell every second. Are you with me? I want you stupefied. And I'm not doing a very good job because the magnitude of this is overwhelming. And people sit around and talk about how that evolved over millions of years by small-scale changes acted on by natural selection. It's foolishness! It's a modern-day fairy tale. Let's keep going. So here you got that RNA being produced, and eventually it's released. Why is it released? Because there's a stop signal, right? And then it comes outside the nuclear pore. See right there, that hole? What did it take, class, to let you in and out of there? Proteins. And so here comes the messenger RNA already properly edited and spliced, ready to move now. Let's watch. It'll be picked up by the ribosomes, which do not look like two ducks hugging each other. They do look kind of like that. It's a glob. And stop right there just a second. Can you see there's the three-letter word, three-letter word, three-letter word, three-letter word that came from the DNA? And you're reading that three-letter word through this machine here. And a bunch of proteins involved. So go. We're going to let it ride a while here, and then we're going to go down inside. Notice all these guys floating around. That's your transfer RNA bringing the proper stuff in here. And here are your transfer RNAs up close. And we're going to stop in a minute here. See that? Yeah. Stop right there. All right. This is the transfer RNA, and this is its tip. That's the amino acid sitting right there on the tip. There's a different transfer RNA for each one of the 20 amino acids. So this is going to show you. Go ahead. Here's another one, there's another one, there's another one, another one, there's another one, there's another one. Stop. I'm just showing you that. There's a specific transfer RNA for each amino acid, and every one of those has a specific that goes with it, or none of it works. So you have to have it all, or the system doesn't work. You have to have proteins to build proteins, class. And you have to have proteins to help the DNA even function. So here we go. Let's keep going. Here's another problem. This is way too slow, class. We're going to go down inside here and look. Watch. Here comes the the transfer RNA. Stop right there just a second. It's got the code down here that's matching up with his code. Up at the top here, he's got the proper amino acids being added to this strand of amino acids. And then the next one comes up, reads the next code, and attaches his one. Let's go. See how it's happening? One at a time? Only problem is that's way too slow. So he's going to try to speed this up for you. He doesn't do as good a job with this one, but watch. See there? Faster! And out the top, you see the beginnings of a hemoglobin protein coming out right there. A hemoglobin is huge. It has lots of amino acids. And inside of it is trapped an iron molecule. That's why it's called hemo, which spreads your iron all over your body. Stop. Now I have one other piece to tell you about this. That's the amino acid strand. You'll notice it's not just a string. It wraps up on itself. And that wrapping, class, is extremely important. Because just because you have a string of amino acids doesn't guarantee you get a functional protein. It has to wrap up into a particular 3D shape. But it goes beyond that. Not only do you have to have this shape here, but this thing is picked up by another, what do you think? Protein, inside of which it has to be shaped further into its final three-dimensional shape, and then it's released into the protoplasm to do the job for which it was designed. And by the way, there are biochemistry textbooks that say it just that way, and then they say, but it wasn't designed. If that wasn't designed, I believe in the man on the moon. I don't mean Neil Armstrong. All right. The chemistry of building proteins in every living cell is sufficient to proclaim there's a great designer. Would you agree, class? I hope so. But we haven't learned half of this. There's so much chemistry we've learned since then. That's tonight. And you don't want to miss it. Because what I haven't told you about is there's thousands of molecular machines helping that happen and lots of other things. Thousands of them. We knew nothing about 20 years ago. And the cell itself is a self-replicating, nanoscale robot. It's self-replicating because it can and does reproduce itself entirely. Did you know that? Your cell completely reproduces it. It's nanoscale because it's very small. Remember, a nanometer is 1/billionth of a meter? And it's a robot because its activities are carried out unconsciously and automatically by precision molecular machines that follow ordinary physical laws, and we know how they work now. And there's no human that's ever made a machine to match any of them, of which there are thousands. You want to know why you get tired sitting there listening to me? You got proteins very busy and DNA busy. Because while you've been sitting there, you have built thousands of proteins and they're busy. It is no wonder that professor of philosophy, atheist, author, and debater Anthony Flew, who for 80 years was an atheist, debated creationists like me. I never debated him. But he did debate a lot of folks, and he was like the standard in the atheist community. When he turned 80 in 2007, he wrote this book called There Is No, a God. How the world's most notorious atheist changed his mind. Here's what he said about himself. It has become inordinately difficult even to begin to think about constructing a naturalistic theory of the evolution of that first reproducing organism. The enormous complexity by which the results were achieved looks to me like the work of intelligence. It now seems to me that the findings of more than 50 years of DNA research have provided materials for a new and enormously powerful argument to design. I say. And Anthony Flew, at 80 years old, changed his mind. Now, he was never convinced of the God of the Bible, but he did become convinced there had to be a God. And it was based on this evidence. So if you're listening to this and you're an atheist, will you please not listen to my yelling? Because I get passionate about it. Because we are told by so many people that just arose by natural causes. Are you kidding me? Look at it carefully and be stupefied. So is it reasonable to believe in God in this scientific age? Lots of my professors would tell you, no, it's stupid to believe in God. No, folks, it's stupid not to. It's more reasonable than it has ever been in the history of man. So I think you can see that that type of a lecture doesn't exactly lead into offering an invitation. But I will tell you how we're going to do this. I'm going to turn to John 14. You want to go there with me? Jerome used that passage today too. John 14 is a good transition passage to bring this to a conclusion. You know the scene here is Jesus on his last days. And he's with his disciples. Verse 1, let not your heart be troubled. You believe in God, believe also in me. I would say to you, if you're an atheist or an agnostic, I hope you'll come to believe in God and that these lectures will help that. And if you do come to be a believer, then I would say to you, the next thing you need to do is learn to believe in Jesus. Well, and Jerome's lessons are going to help you do that because there's abundant evidence that he's the Son of God. In my Father's house are many mansions. If it were not so, I would have told you. I go to prepare a place for you, and if I go, I will come again and receive you to myself, that where I am, there you may be also. You see, the most wonderful thing about believing in Jesus is it has eternal consequences. Because he's coming back, and all those that are his are going to go with him, and you're going to be with him forever in the most wonderful place you can ever imagine. And where I go, he says, you know, and the way you know. And Thomas said, Lord, we do not know where you're going. How can we know the way? Does that sound like me and you? And Jesus said, I am the way, the truth, and the life. No man comes to the Father except through me. If you had known me, you would have known my Father also. Atheists or agnostic, you want to learn about God in more grand detail than you'll ever learn? You need to study Jesus because he is the image and the perfection of God in the flesh. And Philip said, Lord, show us the Father, and it's sufficient. He said, have I been with you so long, and yet you've not known me, Philip? He who has seen me has seen the Father. How can you say, show us the Father? Well, there's two answers to that. We can show us the Father in the natural world, but not like it's shown in Jesus. So the second answer is, you want to really get to know the Father in heaven? You get to know the Jesus that Jerome presented to us earlier today. And our tradition in churches in the United States is we don't want to leave a service like this without offering an opportunity for someone to become a Christian if they're ready. So even if I preached on a totally different subject, it's okay. And our tradition is we stand up and sing a song. Isn't that what y'all do here? Well, I want to just tell you, if you go to Romania, where we lived for 2 years, the churches over there don't do that, so don't have a heart attack. That is a tradition. It's not required. It's found in the book of Jude, chapter 3, which doesn't exist. It's a tradition. Is it a good tradition? I think so, because if you're ready to obey, maybe you need a little encouragement. So we're going to sing a song, and we're going to stand up, and if you're ready to say with your mouth, I believe Jesus Christ is the Son of God, we're going to accept that as coming from your heart, that you mean it. And we'll stop everything. We're not going anywhere till you're immersed. I assume everything's ready back here. In water, because that's what Jesus said, isn't it? He who believes and is baptized will be saved. So we're not leaving until you've been baptized, if you're ready. Come while we stand.